Hello, everyone. My name is Grace Wegman, and today I'm going to be talking about the oncogene-induced DNA damage model for cancer development, specifically authored by Halzanitis et al. In 2008. And again, my name is Grace Wegman. So various terms to know, the first being oncogene, which is a gene in which at certain circumstances could potentially transform a cell into a cancerous tumor cell. The next one is DSBs, which is a double strand break, specifically within DNA. P53 gene is essentially a gene that makes a protein found in the nucleus of cells and plays a specific role in both cell death and cell division. Mutations to this particular gene can actually lead to various tumors that could eventually turn into cancers and essentially spread throughout the body. Another word is senescence, which is very similar to apoptosis, except instead of cell death, it just ages and stops the division of the cell. Neoplasia is essentially new growth, and it refers to new tissue composed of cells with essentially the capacity to be passed down, through generations and essentially grow beyond its normal confines. ARF is alternative reading frame, which is also a tumor suppressor protein. So specifically, as we all know, cancer itself is the uncontrolled proliferation of cells that can either kind of be at the start, benign or malignant. And the new growth of this tissue can be fairly localized, which will eventually, again, go uncontrolled and turn into cancer, which will then invade host tissues. So that's why in important goals of cancer research, specifically the molecular side, is to really understand the genetic basis of cancer and to explain its particular progression. So within that, in regard to DNA damage, DNA DSBs are actually the greatest threat to cells, specifically because these DNA DSBs were thought to actually be incompatible with cell proliferation. So, however, recent experiments actually suggest that both precancerous lesions and cancers have activated oncogenes that create a stalling or collapse of DNA replication forks, which will again give rise to DNA DSBs because of the unchecked proliferation, given there essentially is a destruction of that replication fork that could allow it to, again, go uncontrolled. So this particular formation may cause the genomic instability in most cancers, given that continued formation of those DSBs. So moving on, in precancerous lesions, these DNA DSBs may activate the P53, which without any mutations will cause basically a ceasing in tumor progression, again, because P53 without any mutations account for apoptosis, because it recognizes the fact that this is a double strand break, which obviously is not a normal healthy cell. So it will cause cell death to occur. So the normal genome doesn't have any of those issues. Whereas if there are mutations that somehow arise in that P53 gene, cancers will eventually develop because the cells will go unchecked and will replicate out of control. So that kind of brings us to two of the main questions. There are multiple questions that will be kind of answered or at least brought up throughout this throughout this presentation, because the research is so new that there's more questions than answers essentially. But with this particular oncogene-induced DNA damage model, it'll actually possibly explain two key features of cancer, which is the high frequency of P53 mutations and genomic instability. So again, because it's relatively new, I figured it would be easier to take the bolded headlines throughout the article and talk about those, because again, nothing is conclusive essentially. So it all requires, multiple research and whatnot. So again, to avoid confusion, I figured this would be the easiest way. So moving right along, with DNA DSBs in cells with activated oncogenes in human precancerous lesions and cancers. So identifying DNA DSBs was really, really difficult until the recent identification of proteins that will eventually be taken up by large chromatin near DNA. This makes it really easy to see double-stranded breaks in the DNA using immunofluorescence. So again, like I previously talked about, the most important binding protein found in cancer cell lines grown in tissue cultures is that P53 gene, because it indicates the presence of DNA DSBs, given P53 is specifically produced to activate apoptosis when a DSB is notified. And again, this protein is not visible in normal cell growth because normal cells would never have DSBs, again, because they're normal, they're healthy. So, however, we've kind of come to realize that the analysis of cancer cells in tissue cultures may not actually be as accurate as once thought. So to kind of go along with that, those cancer cells, the cancer cell lines with the highest number of P53 genes had actually an increase in P53 mutations. And this could indicate a particular selection for P53 inactivation, given there is no cell death. So the DSBs, again, can go unregulated, unchecked, proliferate, and result in cancer. So again, it's selecting for an inactivation of it because if P53 were actually activated and not mutated, it would allow for that cell death to essentially be essentially arise and kill off the cell with the DNA strand break. So again, because this is relatively new research and studies, there are a lot more questions than answers, but to kind of shed light on future research and kind of how to go about this, there are a handful of experiments that have been done that I'll kind of go into throughout this presentation that have been conducted on both precancerous and cancerous lesions of human patients. So specifically to identify DSBs in both precancerous lesions and cancerous lesions, we'll start with precancerous ones. So specifically in precancerous lesions, before there are any P53 mutations, the presence of DNA DSBs can actually be identified through the inactivation of the various checkpoint proteins during DNA replication, such as ATM, 53 binding protein 1, CHEK2, and then P53 itself, again, all non-mutated. Whereas in cancerous cells or cancerous lesions, the primary indicator of DNA DSBs is actually the DNA damage checkpoint pathway that's been compromised by those P53 mutations. So where cancerous cells, the DNA DSBs is again specifically those P53 mutations. In precancerous ones, it's the other DNA replication sites before P53 mutations can occur. So moving right along to the next headline. Oh, I lied. So really quickly, basically, the main takeaway of that, or of this essentially, is that the presence of DNA damage was actually thought to be specifically distinguished between precancerous lesions and cancerous ones from those normal tissues. But it actually gave rise to a bigger question as to how the DNA DSBs specifically arose within those either precancerous or cancerous lesions. So this is why the presence of activated oncogenes is essentially now being heavily researched to hopefully provide evidence to, again, the above question, given oncogenes can create both DNA DSBs and the DNA damage response. So now moving on, the next headline is a tumorigenesis barrier in human precancerous lesions. So the main question with this one is that is apoptosis and senescence present in precancerous lesions, and how do they subside during cancer progression when P53 mutations are present? So there were 3 different tissue samples that were essentially collected and investigated at normal precancerous and cancerous cell levels to try to kind of answer this question. So the 3 different tissue samples were lung, melanoma, and colon. And as you can see in this figure right here, again, we have the lung, melanoma, and colon. This is the normal cell, this is the precancer, and this is the cancer cell. So this green part right here is the proliferation index. This orange line is the DNA damage response index, and then the purple line is the apoptosis or senescence index. So essentially what happens is the general pattern, as you can see, is that there are incredibly high levels of apoptosis and senescence during these precancerous cell essentially stage in development. And that gives rise to the conclusion that there is a specific tumorigenesis barrier in this particular stage of development that will eventually, be broken to create and give rise to cancerous cells, as you can see over here, which creates a decrease in the DNA damage response and the apoptosis and senescence. So what's interesting about this is that the original thought was that precancerous lesions were actually less aggressive because of the fewer oncogenic mutations that were present. However, these models in these various cells, albeit that they had, different reactions, but all gave rise to very similar conclusions. So it relatively proves that the precancerous lesions are actually less aggressive due to the presence of this tumorigenesis barrier essentially during this DNA damage checkpoint. And it's really interesting to see in the colon tissue sample specifically because of the, in these precancerous adenomas, they actually have a lower proliferation rate or index than the normal cells do, which is incredibly weird because as you can see in the melanoma and the lung, it's quite the opposite, or it relatively stays the same. So it's very interesting to kind of figure out. Again, moving on. So with the next headline is the DNA damage checkpoint as an important mediator of the tumorigenesis barrier. So there is an incredible amount of evidence specific for the p53 gene as a very, again, key gene that creates proteins in the tumorigenesis barrier in precancerous lesions, because again, apoptosis and senescence are very p53 dependent. While moving beyond that barrier, that tumorigenesis barrier to full-blown cancer is when you get those p53 mutations that again cause that cancer. So there were various tumor model or mouse tumor models, cultured primary cells, overexpressing oncogenes, analysis of colon and bladder precancerous lesions, specific immunohistochemistry that all suggest that the p53 is activated by the DNA damage checkpoint in cancer patients. However, again, because this is so new, as I keep reiterating, there are lots of conflicting and contradicting evidence to kind of dissuade the idea that DNA damage checkpoints are an important barrier to tumorigenesis. And that leads to a bunch of other questions that which I won't get into to spare you guys the confusion. However, I do think it's really important to note that there are two current studies specifically looking at tamoxifen regulated p53 protein within ARF null mouse model that actually concluded that the tamifixin regulatable p53 actually requires both tamifixin and a DNA damage signal to create that apoptosis, which is relatively consistent with the precancerous cells in mice having those DNA DSBs, again, because once that p53 is activated and like mutated, that DNA damage checkpoint is essentially also activated as well to create, unfortunately, cancer. So moving along to one of the last headlines is DNA replication stress underlies DNA DSB formation and genomic instability in human precancerous lesions. So a key question kind of based on the results thus far relates to the mechanism by which the activated oncogenes are constantly essentially creating these DNA DSBs in both human precancerous and cancerous lesions. So it was kind of seen that oncogenes could induce DNA replication stress in humans or in human precancerous lesions that lead to the formation of those DSBs. So the DNA replication stress can specifically be started by the increase in number of stalled DNA replication forks, again, because it allows for cells to go unchecked at various specific chromosomal loci. Loci create these microdeletions and essentially chromosomal rearrangements due to the formation of these DSBs to try to kind of counteract the effects that actually give or given the replication fork collapse. So expression of oncogenes in certain cells also leads to DNA replication stress. Again, because of the destroyed DNA replication forks, it leads to those DNA breaks, DNA strand breaks. So there is a prediction that the oncogene-induced DNA damage model is that this genomic instability in both human precancerous and cancerous lesions is actually just induced by the oncogenes themselves in very early stages of cancer. So without, all the hoopla, just the oncogenes themselves. One mechanism to try to prove this was telomere erosion. Another mechanism was actually the permanent increase in genomic instability was essentially induced by mutations in genes that are known as caretaker genes that were essentially specific to the function to try to preserve genomic integrity. So these are like your DNA repair systems or the cell cycle checkpoints. And kind of in regard to that, most cells have chromosomal instability or CIN in general, which can occur specifically due to these DNA DSBs because again of this DNA replication stress that's kind of put upon it. So it is quite possible that DNA oncogene induced DNA damage rather than just the inactivation of these caretaker genes. It actually may cause the CIN or the instability in essentially human cancer. So oncogenes themselves may have the ability to do such a thing. It's just not, it needs to be more researched essentially. And genomic instability itself is really, really important for cancer development essentially, because without it, you wouldn't have any mutations that would be acquired by the genome in order to actually create cancer. Because again, cancer is a mutation of that, well, one of the pathways is a mutation of that P53 gene, which, stops apoptosis, allows the cells to proliferate uncontrollably to cause cancer. So without mutations, there is no cancer. It's a normal, healthy cell. Having said all that, though, it's still very unclear as to whether genomic instability induced by DNA replication stress has a role in cancer progression. So again, further research would need to be done to essentially prove that. Moving on to conclusions and future directions. So again, this is all very new, inconclusive, roughly data, research, whatnot. So this oncogene-induced DNA damage model that's been obviously mentioned throughout the presentation for cancer development and progression is very good to shed light on, various aspects of cancer, such as progression, genomic instability, and the tumor suppressor role of that P53, gene. However, like I mentioned, the model is still very new, very green, is not perfect. So it definitely needs more definitive research, given there are still a lot of questions that have been left unanswered and a lot of unclear mechanistic purposes. However, within that, as far as the future goes, this is a great jumping off point. So I see new methods for diagnosing precancerous lesions on the horizon, including the potential for immunohistochemistry assays for DNA damage responses in a various clinical setting, and the development of various new cancer treatments and various therapies. So like I said, the future is incredibly bright, but it's also incredibly unclear because it's very, very much dependent upon the research that we have available and whatnot. But I think, again, for the future, it's very, very bright. So these are my questions at the end of my presentation. These are my questions that will hopefully guide you guys through my presentation. Hopefully this helps. Again, this is my reference from the article. And I really appreciate you guys listening. Thank you guys for taking the time.